Well, thank you again. That was a great talk, Katya, or inspirational. I don't think I'm going to follow it up, but what I will try to do is pick up where I stopped over the last couple hours ago and try to share with you some of the data that we have generated over the last numbers of years about what is happening when a subject is transitioning from acute to chronic pain. My lab also, over the last, I don't know how many years, I'm not going to tell you the number of years, we've always been doing animal studies at the same time as human studies. And the idea is always to back translate from what we learn in the human condition and to test it mechanistically in animal models. And there's a big debate in our field, again, as to the validity of a lot of the animal models that have been tested and studied over many, many years, as to their usefulness. In many ways, they have not translated into new clinical efficacy outcomes. And as a result, one tends to blame the model as opposed to whatever we have asked, in a sense. So I am interested in the validity of animal models and how much they do correspond to the human condition and how we can use them in further understanding these conditions. So I'm going to spend some time discussing that as well. So let me begin. So I showed you a lot of evidence that the brain is changing, that the brain is not the same in acute versus chronic pain. The anatomy is not the same. The physiology is not the same. But this is the critical question. The question is, you all know that patients with similar injuries, the vast majority of them get better after the injury. Only a small proportion of them develop chronic pain. So what is the difference between the two? What controls the transition into chronic pain versus the reversal back into health, right? In acute back pain, which happens to all of us at any given time in our life, 95% of patients become back to normal within weeks from the injury. A very small proportion of them continue to have chronic pain. What is the difference? That's the question I would like to address over the next hour. So again, I'm coming back to my favorite picture of Descartes' idea of sensory representation. Again, I mean, this is basically our history of pain research, and what is our expectation? The idea is if the nociceptive input from the periphery up to the cortex is the simple pathway with which we are feeling pain, then our expectation would be very simple. Anything that happens here or in the spinal cord, if it's sustained, it becomes chronic. And in that sense, one can explain away all of the brain reorganization as simply a consequence of the persistent pain, right? Rather than a causal relationship. So yes, the brain is reorganizing because the subject is suffering with their pain. Still, the stimulus is in the spinal cord, or the drive is in the periphery, and the brain is simply coping or reorganizing in relationship to the nociceptive input. So even though we see all of these changes, what is the drive? Where is the critical component? What parameters critically determine what's going to happen to the patient? So again, this is my definition, my new definition of what is chronic pain. And actually I'm going to spend the rest of the hour essentially trying to deconstruct this idea and show you both animal and human data along those lines. Essentially, again, that the brain limbic properties, the learning events in the brain limbic properties, their initial state, all of those things are the controlling parameters that determine what's going to become a chronic pain brain. And in fact, we now sort of think of chronic pain as composed of multiple stages, four different stages for chronic pain. A predisposition, an injury event, right, a transition, and maintenance. And in a sense, we think that each one of them has its own properties, and the interaction between them is what's going to determine the outcome. Again, from a classic viewpoint, one could simply say all of these things are basically related to the nociceptive afferent input. Right? You test for hypersensitivity in the patient, you ask them, did they have pain in the past, and then you assume all of those things are the primary determinants for someone becoming chronic pain. I don't think that's true. So let me show you what we think is happening. In fact, so this is a hypothetical model we have initially put forward a number of years ago before we had the evidence. But essentially it says that the nociceptive input interaction with the limbic brain is going to reorganize the brain itself, and that reorganization will change the brain's perception of that stimulus itself from a nociceptive sensory input into an emotional self condition. That's the overall general model. And in fact, what I want to say is that this model is going to have components of which would be part of the predisposition. The inciting event would be the driving nociceptive input to this system, and how the system reorganizes in the transition would be, again, part of the model. Really, what happens at the maintenance level, I think there are probably very specific mechanisms we have very little idea about, and that remains to be determined in the future. So, a number of years ago, we decided to do this crazy experiment, actually take basically acute back pain patients and follow them over a 1-year period and see what happens in their brains. This was I mean, at the time, even though we got funded on this idea, if anybody had asked me how we were going to do this study, I would have said, I have no idea, we just have to see what comes out. It was essentially a data set that we collected over a 5-year period. We collected about 3,000 brain scans in these subjects, and we had no idea if anything was going to work or not. We assumed that if we include into the study subjects who had a relatively constant duration of pain, more than 4 weeks, less than 12 weeks of back pain, and if the pain was present, had an intensity of more than 5 out of 10, that would give us enough pain that enough of them would then transition into chronicity, and then we can untangle what is the difference between the two groups. So that's the general idea. We could, and to also discount the past pain history as part of the situation, we also excluded subjects that had back pain for over the last 1 year. So these are subjects that have had no back pain at least for a year, and yet have had an acute episode of back pain, which is fairly constant and fairly persistent. And then we bring them into the lab. We simply do a series of scans over multiple time periods and analyze the data. Both functional imaging and anatomical imaging in these subjects. So what is the role of the brain in all of this? That was the overall simple question that we were trying to ask. So this is sort of, again, the design of the study. Subjects enter in this subacute state in that their pain is more than a couple of weeks sustained. And then we scan their brains 4 different times over a 1-year period. Now, in fact, we've extended this to another 2 years. So we have data all the way up to 3 years from the initial event. And then we call these guys as subacute back pain patients. And then some of them are going to have pain sustained over that 1 year. Those are people we will call transitioning into chronic pain, while others we will call them recovering. We call them recovering by a very simple definition. If their pain decreases by more than 20% over that 1 year period, we simply call them recovering patients. And so by design, we have brain parameters at time 0, and we have brain parameters over the whole 1-year period. So we can distinguish between consequences of transitioning into chronic pain versus predictors. So anything at this time that can predict the outcome a year later, in a sense, by definition, is a causal biological parameter that controls the transition to chronic pain. On the other hand, things that are happening here in the persisting population, but not in the recovering, those are the processes that we will call them as creating the chronic pain brain, in a sense. Okay? So here is the behavioral outcome. These are at time 0. All the subjects have pain by design around 5 out of 10. This was the criteria for entering into the study. And they all have pain duration of equivalent time as well, because by design, by the way we've designed the study. And we were lucky in a sense that 1 year later, about 50% of the subjects don't change their pain at all, while the other 50% continue to decrease in their pain. And even though our criteria was just 20% change, note that over the whole population, the change is more like 300%. Okay, so there's a massive, so the patients who are recovering look like they're really getting healthy, becoming healthy subjects. So even the simple behavioral parameters in this data set are interesting, and I want to share some of it with you. Because there's all this, you know, Katja also mentioned this biopsychosocial models of pain where anxiety and depression or fear are all thought to be important parameters. It's not obvious whether they are consequences or causal relationships, so the behavioral parameters themselves are informative. And what we see, in fact, is that at least at time 0, at visit 1, the two groups, the persisting and the recovering group, are essentially have all of the pain and anxiety and depression parameters are all matched. That's good for us. Right? So we're starting with a matched population. 1 year later, the persisting pain population, not much changes. They're all exactly at the same level of anxiety and depression and pain. Those parameters have not changed at all. The only thing that's changing is that in the recovering group, all these parameters are decreasing. So the subjects who are getting better, their clinical measures are all improving. The subjects who are not getting better are essentially exactly at the same level as they started initially in the study. Okay, and so one question is, how is the brain activity changing in time? I told you in the earlier study that when we look at the brain in chronic back pain patients, we see essentially only a prefrontal brain activity when they are already in chronic pain, if they have lived with that condition for many, many years. So here we have a population of subjects that some of them are recovering and some of them are persisting. What's changing in their brain activity when they actually rate their pain the same way that we did initially, their subjective pain activity. So at time 0, first of all, the two groups show very similar brain activity. And the activity that they show, in fact, matches acute back pain, not back pain, acute thermal mechanical stimulus related activity. So the brain activity at the initial state, even though they're rating their pain, their subjectivity, it actually matches very closely to the meta-analysis data that I showed you, or it matches the pain signature that Tor Wager, for example, generates. All of those things we call them the acute pain brain sort of signature. That's exactly what these patients are showing. Over the year, the recovering subjects, their pain is going down, right? Their pain is decreasing. And in fact, by the end of the year, we don't see any brain activity for their rating of the pain. That's not surprising. There's not enough pain. And we don't have enough signal to detect anything anymore. The persisting patients are the more interesting in that the brain activity does shift in time, and it shifts away into exactly what I had showed you cross-sectionally earlier. In a sense, 1 year later, the medial prefrontal cortex and the bilateral amygdala are the only areas left that are activated with their pain. Even though the subjects McGill pain questionnaire, anxiety and depression, all of those things have not changed. Their brain activity has dramatically shifted from sensory areas to emotional areas of the brain. And how is this happening? It's happening because their brain is changing in time, right? And so let's look at some of that. So let me now show you what's happening to the anatomy of the brain in these patients. Again, these are the subjects who are persisting, their pain is not changing. Here are the subjects where their pain is decreasing in time. I have now included 17 healthy subjects that we have followed exactly the same way as an internal control. We have, again, scanned them multiple times over a 1 year period. And in this panel, I'm actually plotting the overall neocortical gray matter volume. How much volume corrected for age and gender and for body mass, body size. How much is your cortical gray matter? And the subjects who are recovering and the healthy subjects are matched exactly. Only the subjects who are developing chronic pain is that volume decreasing in time. It's decreasing continuously as the pain is persisting, the gray matter volume is decreasing in time. Okay, so this is a global measure of the overall brain. We can then ask the question, what brain areas are changing? And that's the next slide. And so if we simply compare the brain regions now that are changing in their gray matter density, we see that in the persisting patients, there are multiple brain areas that are decreasing. In the recovering brain and the healthy brain match each other, so that in the recovering subject, at least we don't see any active reorganization of the brain. Only in the patients where the pain is persisting is the brain anatomy changing in time. And specifically, the brain areas that are most interesting is the somatosensory cortex, insula, and nucleus accumbens. And you can see in all of these cases, the healthy controls and the recovering subjects show no change at all, but only the persisting patients show a decrease in gray matter density. And okay, sorry, again, my slides are a bit messed up, so I'm trying to see what I have. So if we look at one of the specific areas, this is the insular area. That's the cortex that was decreasing in the persisting subjects. We can show that not only is the anatomy changing, but the functional properties of that area are changing as well. And in fact, both the gray matter density, the functional connectivity, and the pain are all interrelated with each other. So in a sense, we see functional and anatomical changes that are essentially recarving the brain, and we define this as now the emerging new brain state of chronic pain. If we look at the nucleus accumbens, we find something different. The nucleus accumbens is this region here, and we look at its connectivity, its functional connectivity. What brain areas is it's talking to? We see that it's essentially talking to only the medial prefrontal cortex. And that functional connectivity is there both in persisting and recovering subjects, and it's not changing in time. Still, that connectivity is correlated with their pain as well. So here is a brain region that anatomically is changing, but functionally is not changing, and functionally at time 0 is already different between the two groups. Okay? And if I just plot that connectivity, that's this accumbens medial frontal cortex connectivity, you can see that at time 0, the persisting subjects versus the recovering subjects, that amount of information shared is different between the two of them, and it's different at visit 1, visit 2, visit 3, and visit 4. So here's a parameter that exists at the time 0, and it's not changing at all. So that's a candidate as a predictor, as a property of the brain that's going to predict the long-term outcome. Now, we've also looked at the white matter properties in these subjects as well, right? And again, these are the subjects that are persisting, and here are the subjects that are recovering. And if we simply look at the white matter, we see a whole number of regions in the white matter that distinguish the two groups from each other. And if I simply take, this is simply saying that there are different bundles of axons, the properties of which are distinct between the subjects who are recovering versus the subjects who are persisting into chronification. And if I simply plot that overall area, fractional anisotropy, essentially a scale of how much myelination or how much axonal branching is happening within those areas, there's a big difference between the persisting and the recovering at time 0 again. Now, again, we have the same information in chronic back pain patients. These are patients on average have had pain for more than 10 years, and control subjects. And you can see that the same trend already exists in these groups as well, in that the chronic back pain patients and the persisting SBPs show similar abnormality in white matter, while the control subjects are somewhere halfway in between. Again, suggesting that this is already a pre-existing condition, that in the population, some of them are going to have a brain like this, and others are going to have a brain like that. And if then they happen to have a back injury, that would then put them at different risk of developing chronic pain. Okay. So if I plot now this fractional anisotropy, this was at time 0. There's a big difference between them. And again, this is a parameter that's constant, and it's not changing over 1 year. So both of these parameters seem like they should be predictors of outcome. So before I show you the prediction, I have to make a little joke, I suppose. And this is, making a prediction about the future is very hard. And the longer the time window is, in fact, it gets harder. I mean, we all know this, especially in science. On the other hand, in this case, when it's chronic pain, in fact, the longer the time window, the more confidence we would have that it is chronic pain. So we want to have it the opposite of that idea. Okay, and this is my tutorial about receiver-operator curve, simply to show you that how we can calculate these things. Basically, if you have two distributions, right, the recovering versus the persisting, the overlap between these distributions is essentially what we are calculating when we plot these ROC curves. And the further apart they are from each other, the bigger are these curves. If we cannot distinguish them, then it would be a straight line. Chance here, moderate, and perfect would be that triangle up there, right? So here is the ROC curves now for the functional connectivity between these two areas of the brain. These curves are for the 3 subsequent visits based on the data at time 0, right? And so, and the area under these curves essentially give rise to the accuracy over which I can make the prediction, the future prediction of outcome. So at 3 months, my prediction is around 68% correct. At 6 months, it's more than 80%, and at 1 year is again more than 80%. So that's a pretty robust result. So I can make 80% correct outcome. And in an unbiased validation data where we only calculate this, we don't do any other thing, again, we end up with 80% correct outcome. So here's a one parameter, a single value of the amount of information shared between two brain areas that gives me 80% accuracy of telling you whether the subject will develop chronic back pain or not. Now, if I combine that parameter with multiple other parameters, how much better can I do? And what does that model end up being? First of all, that parameter by itself has an odds ratio of 5. That's a huge number in the sense that that says that if that number is high, that subject has 5 times higher probability of developing chronic pain than a subject that has a low number for that value. Okay, so its predictive value is very large. It's much larger than, for example, the genes that we have identified for chronic pain. If I combine that with 2 other parameters, my odds ratio, in fact, becomes much larger. Now, the brain parameter becomes 13. On the other hand, what we can show is that treatment of any type, as long as the subjects receive some sort of early treatment, it is protective. So that, that treatment becomes a significant parameter only in the model that includes the brain. Without the model, that treatment is not effective. Okay, so in a sense, that variance can be explained only in combination with the brain itself. This is, again, the ROC curve now for the white matter properties that I showed you earlier. In this case, again, 3 months, 6 months, and 1 year, the accuracy is even better. Look, at 1 year, I am 100% correct in identifying who's going to become a chronic pain patient versus not. A single parameter, the brain white matter property, explains all of the variance of the risk for pain chronification. And again, in a validation cohort, we end up with about 80% correct. Okay? Now, we've analyzed this, we continue to analyze this data set, and it's a large data set. As I said, there are like about 3,000 brain scans in them and a large set of parameters. And we've now been looking at subcortical structures. And I'm just sharing you some of the data that comes out from just the volume, the hippocampus volume and the amygdala volume. Both of them are smaller in the subjects that persist to become chronic pain patients. They are smaller at time 0, and they are smaller 1 year later, and now we know they are smaller 3 years later. So these are, again, we think, parameters that these subjects come with to start with. These are parameters that put them at risk. And simply, if I just take the hippocampus volume by itself, I'm 68% correct in identifying who will become a chronic pain patient. I'm about 80% correct just with the amygdala itself. Okay, so we've also now looked at a number of genes, whether we can find some correlations of genes with some of these parameters, and we can. I'm not going to belabor the details. Essentially, we can find specific genes that correlate with hippocampus and amygdala volumes, for example, and in a sense indicating that some of these parameters are at least genetically predetermined. All right? The model here I am showing you is in an effort to show overall how much can I predict of the intensity of the pain 1 year later. So far, I was simply saying who is going to persist or recover. If I simply take these 4 parameters, pain intensity at time 0, at entry into the study, and these 3 brain parameters all together, I can explain 80% of the variance of the pain 1 year later. Okay? 50% of that comes from the intensity of the pain at time 0. So the pain at entry determines how much pain I will have 1 year later. But the brain parameters are the ones that determine my risk to developing the chronic pain. So the nociceptive input is important. The nociceptive input is determining the state 1 year later. But it has no predictive value of who will develop chronic pain versus not. Okay, I don't know. All right, so that's the human data. I want to spend the next few minutes just give you some of the data that we have been developing in animal models. The point here is that we're literally trying to find mechanistic equivalence where we can in much more detail identify what is happening in the brain that we cannot capture, obviously, in humans. So we recently have done some, again, similar sort of longitudinal studies in animals, in rodents, where we give them a neuropathic injury and look at their brain reorganization in time. So that's sort of a longitudinal animal model study. I'll show you some data from that. And some of the data about the role of the hippocampus and the role of the nucleus accumbens and how those regions of the brain are involved in now animal models of chronification of pain. So here is the data. This is resting state brain activity in rats under anesthesia. Okay, these animals have had injury, a peripheral nerve injury, and then we scan them 5 days later and 28 days later after the injury. And this is the brain network as an overall network and its reorganization. At 5 days after the injury, this blue line is the only difference between sham-operated animals and the neuropathic animals. 28 days later, all these blue and red lines are different in the neuropathic animals from the sham-operated animals. So the brain is reorganizing in time at a whole brain scale, and we can quantify what are the areas that are reorganizing. And essentially what we find is that brain limbic areas in relation to nociceptive regions are the main areas that are changing. So that's very nice. It sort of validates that, in fact, what we're seeing in humans, we can actually begin to translate into animal models as well. I've showed you this data already. I'll skip over it. Now, if I simply look at nucleus accumbens and look at its functional connectivity, again, what essentially we see, I'm not going to spend much time on it, is that 28 days later, that its connectivity has changed, but 5 days later, there is no change in its connectivity. They're also related to that now dopamine receptor changes in nucleus accumbens that happen essentially at 28 days, but not at 5 days. And those changes are actually correlated with the tactile allodynia that these animals show. So at 5 days, the tactile allodynia is there, but we don't see the brain parameters related to it. 28 days later, both the dopamine receptor changes and functional connectivity changes are now correlated with the behavior itself. And in fact, if we block activity in nucleus accumbens by lidocaine injection locally over a 2-week period, we can decrease the tactile allodynia itself. So this is a very high level sort of manipulation locally that's changing a local spinal cord reflex essentially for pain. We've also now been doing patch recording from neurons in nucleus accumbens. These are individual neurons that we isolate, and they are labeled by green or red, depending on the type of receptor is sitting on top of them. So we can identify D1 versus D2 receptor types and look at their excitability changes. And we can see that within days, the D2 receptor type neurons increase in excitability only in the shell part of nucleus accumbens, and that's preserved over 28 days as well. So there is reorganization at the synaptic level now of information processing within the region. We're very excited about the role of the hippocampus in chronic pain. Why? Well, hippocampus is the brain's primary region where learning, reinforced learning is happening, right? And if learning is part of the chronification of pain, then the hippocampus should play an important role. And we have shown, for example, if we, animals with neuropathic injury, if we test them on different learned behavior, they're normal on cue extinction type behavior, but they're not normal on context extinction. And context is a hippocampus-dependent paradigm. In a context test, basically these animals are unable to forget their learned fear conditioning. Okay, so they're unable to extinguish it. That directly implicates the hippocampus in these things. And if we actually look at what is another important peculiar property of the hippocampus is it's the only area in the brain where in the adult brain there is neurogenesis, right? Adult neurogenesis only happens in the hippocampus, and we think it is part of the hippocampal learning process. And in fact, hippocampal neurogenesis is dramatically decreased in neuropathic injured animals. And if we block neurogenesis, at least transiently, the animals are unable to develop neuropathic pain. So we think that hippocampal learning is a fundamental process of the transition from acute to chronic pain, which in turn will propagate throughout the limbic system and reorganize the rest of the brain. Okay, so this is my summary slide. What I tried to show you is that there are multiple very simple brain parameters that are predisposing factors to becoming chronic pain, and they explain the large, almost all of the variance of transitioning from acute to chronic pain. So the nociceptive signal does not come in there, for example. What comes in are brain limbic regions and their connectivity and their interaction, right? The white matter properties is the interaction between them. The functional connectivity is the extent to which those regions are interacting with each other. And the volume, again, says something about the synaptic properties of these areas. All of these essentially explain away all the variance of the transition from acute to chronic pain. The transition we still think is driven by this functional connectivity, which in turn is changing the synaptic properties of the whole limbic system, and that's driving the anatomy of the brain to transition into a new brain state. That's only happening in the ones where the pain is persisting, but not in the subjects who are recovering, which gives rise to the